So welcome everyone to the ACNM non-CE webinar brought to you by the American College of Nuclear Medicine and the Nuclear Medicine Residents Organization. Today's topic is, does the term PSMA create a gender bias? Today's speaker is Dr. Marigdalia Ramirez-Fort. Dr. Ramirez-Fort earned her undergraduate degree at Lehigh University and medical degree at Ponce Health Sciences University. Her general surgery internship was completed at the University of Pittsburgh Medical Center. Since then, she has trained in radiation oncology at the SUNY Downstate Health Sciences University and Medical University of South Carolina, jointly with a postdoctoral research fellowship in urological oncology at Weill Cornell Medicine. In July 2022, Dr. Ramirez-Fort commenced residency training in nuclear medicine at the University of California, Los Angeles Veterans Affairs Program, where she became the first resident from the program to be awarded the ACNM internship. To date, Dr. Ramirez-Fort authored more than 70 peer-reviewed articles, abstract, and book chapters, and has served as the lead editor for a textbook on human papilloma virus for Karger Publishers. Now she serves as co-editor for the Frontier in Medicine Science series issues, Women in Science, Nuclear Medicine 2024. She has experience in running approximately 30 investigator-initiated or pharmaceutical-sponsored clinical trials as sub, co, or principal investigator. The latter portion of these trials involved the development of the theranostic target PSMA encoded by the FOLH1 gene. Throughout her training and career as a physician scientist, she has been involved in a wide range of preclinical and clinical research, including studies involving the effects of different wavebands of electromagnetic radiation on cellular and organismal level. With her longtime professor and mentor, Dr. Sir Christopher S. Lang, they have defined the pathophysiology of human herpes virus reactivation in radiotherapy patients and defined the organ at risk for development of sexual toxicities associated with prostatic irradiation, and more recently as president and chief executive officer at BioFort. Her investigational focus involves the development of patented lesional microdosimetry, fractionization, and dose optimization for the radiopharmaceutical targeting of a variety of cell surface receptors. Dr. Ramirez-Fort?
Hi. So thank you for the introduction. So these are my disclosures. What I am going to, share with you is not just based on my lifetime of knowledge, but it's also based on my professor's lifetime of knowledge and his professors and his professor, which was actually George Hevesy, that was awarded the 1943 Nobel Prize in Chemistry for his intellectual contributions in our understanding of biological applications of radioisotopes. So boom, the atomic bomb in 1945 really triggered an enormous global national investments in radiation research. This really made us feel a Darwinistic crisis around the world, where the question was, how do we survive? And can we use this energy for any peaceful purposes? After that, in 1956, Puck et al. Published the first dose-response curve for the survival of reproductive integrity, more generally known as the cell survival curve. So it's important to understand what reproductive integrity means, and this means the ability of a cell to reproduce itself ad infinitum. So that's cancer, a cell that keeps dividing. Cell integrity or clonogenicity means the ability of a cell to make a clone or macrocolony, which is meant to undergo 5 or 6 mitotic generations. So in total, approximately a 50-cell colony. The survival curve and the publication of the first survival curve was really important because it allowed the in vitro calculation or quantification of surviving fractions of clonogenic cells from different acute radiation doses. Specifically, that allowed us to be able to calculate the doses needed for a cure, a tumor cure, or calculate the doses resulting in normal tissue toxicities. So here on the left is a figure from Puck et al.'s publication in 1956. On the left, figure 1 and 3 shows a plated cell colonies for different days or different amount of time being plated. And then on the right, we see the same plated colonies after 300 centigrades or 3 grays of radiation. The point is that as the dose goes up, the number of colonies goes down exponentially. And that is depicted as a straight line on the semi-log plot, which I will show you again shortly. All right, so here we see the semi-logarithmic plot. So the y-axis is a logarithm of the surviving fraction of cells. And the x-axis is the linear radiation dose. And so we use a logarithmic scale for to evaluate the surviving fraction of cells, so the processes of cellular death and survival, because it allows us to see survival at very low surviving fraction. So it allows us to see very small changes, which we wouldn't otherwise be able to see on a linear scale. All right, so what we see is on the straight line is an initial slope, a curvature away from it, which is called the shoulder, and followed by a straight line exponential. The shoulder is determined by the kinetics of double-stranded DNA breaks, their induction and their repair, and the terminal slope, which is a straight line, is indicative of an exponential process. It enables us to, if we didn't have the available data set, which we have here, it would enable us to extrapolate all the way to the right and for very large doses of radiation. So how does fractionation affect the survival curve? If one fractionates the dose, it turns out that the shoulder is regenerated with daily fractions. The second shoulder starts at the surviving fraction of the first dose on the first curve to generate the survival curve for 2 doses, and etc. And if this process is repeated multiple times, we see that we can draw a new exponential that connects the surviving fractions from each dose to give the effective survival fractionation curve for that particular fractionation regimen. And here is a very simple multi-fractionated dose of 4 doses. This curve is based on almost all clonogenic cells. So that's not normally what we see in real life. Real tumors have a heterogeneous population of cells consisting of transit amplifying cells and cancer stem cells. And so in 1967, Lange, in his thesis, demonstrated that the repopulation probability of a tumor or normal tissue is based on the repopulation probability of its respective stem cells. And to the extent that not all tumor cells are clonogenic, like your normal real tumor sample, then the number of cells used to generate the dose response survival curve is not the number of tumor cells plated, but the number of the cancer stem cells within the tumor. So that means that lower doses are needed for cure than what would be derived from Puck's survival curve, which is a good thing. So a year later, Lange and Gilbert developed a model, the LNG model. That was the first to relate the normal tissue stem cell survival curve to the organismal survival curve, forming the basis of our current day understanding of normal tissue radiation toxicities. First, the Lange and Gilbert model showed that in planarians, the slope and intercept of the flatworm's LD50 curve are defined by the slope and intercept of their underlying stem cells and the number and repopulation probability of those stem cells. Interestingly, I came across this paper and, well, I didn't know until I saw it, but apparently planarian expressed PSMA. So back to what I was saying about the LNG model, when we do a 2-step application of the LNG model, the same was shown for irradiated mouse gut lethality syndrome, where the mouse LD50 curve was defined by the survival of gut crypts, which in turn was described by the survival of the approximate 8 stem cells per crypt. And this 2-step procedure led to the concept of functional subunits. The same way that the LNG model was able to predict toxicity in radiation limiting organs, it can be applied to any organ system, for example, the kidney or hematopoietic cells, to calculate radiopharmaceutical induced tumor cure and or dose-limiting toxicities. In 1973, Longo published his discovery that DNA double-stranded breaks in mammalian cells are repairable. This was a very important discovery, and it took several years afterwards, in 1986, for Ostashevsky and Longo to develop the DSB model showing that stem cell survival could then be understood at the molecular level in terms of the kinetics of DNA double-stranded break induction and rejoining. The DSB model demonstrated that the initial slope of the cell survival curve and its shoulder, so the shoulder region that is used to reconnect when we fractionate, that region is determined by double-stranded break rejoining kinetics. And the DSB model correctly predicted cancer and normal cell survival curves from this DSB repair kinetics data. For completeness, this is the complete equation. It has been shown that DSB rejoining and consequent reduction in illegitimate rejoinings are responsible for dose fractionation induced reduction in cell lethality and normal tissue toxicity. So this shoulder is what allows for normal tissue to repair and reduces toxicities with radiation. And that also applies to radiopharmaceuticals. This was a pilot study that was done, and you can see already in the background of the paper, it's at a modest dose fractionation of lutetium J591 to 2 doses had less myelosuppression per similar cumulative dose. The hypothesis of this pilot study was that additional dose fractionation would allow for a higher cumulative dose, potentially with less toxicity and more efficacy. And we did see that we could achieve a higher cumulative dose with less toxicities. Back to the DSB model, what else it predicted that was really important for clinical nuclear medicine and radiation oncology is that the DSB model can show us how much time is needed between infusion fractions of radiopharmaceuticals to allow for normal cellular repair to an acceptable toxicity. So all of these aforementioned discoveries and derived models are the cornerstone to our current understanding of external beam radiation fractionation, dose per fraction, and dose rate. Moving on to my work with PSMA. In 2014, I joined Dr. Neil Bander's lab. He was recently named one of the founding fathers of PSMA technology. He created the first antibody to the extracellular portion of PSMA. From the very beginning of my reading when I joined the lab, I noticed this discrepancy in the literature where there was this term being used, prostate-specific membrane antigen, which implies specificity to the prostate, but that just wasn't so. And so I started to question in the back of my mind, why are we even calling it prostate-specific membrane antigen? Another thing in the literature is that there were all these different names used to describe the same enzyme. So that was pretty confusing. Then my first project in the lab involved inducing PSMA expression on HUVEC cells. So what we did is we took substrates from solid tumors, and what we showed is we were basically able to take substrate, so waste media from a whole bunch of different non-prostate cancers, and all of them induced PSMA expression in undifferentiated endothelial cells. And then we showed that after we induced the expression of PSMA, those HUVEC cells or neoendothelial cells could effectively endocytose PSMA and whatever was conjugated to it. So this is an immunofluorescent video of the cells endocytosing PSMA. I got really excited about this project and what I had read, and I had been doing research in the area of melanoma since 2007 and Merkel cell carcinoma since 2011. And so I asked Dr. Bander, can I look at PSMA in non-prostate cancers? And he said, yeah, as long as you do it on weekends or after hours and you buy your own supplies. So I said, okay. All right, so my first project was a melanoma, and I replicated what the lab had already done in 1997 in terms of just using immunohistochemistry to demonstrate that the neovessels express PSMA. Then we identified that primary and metastatic melanoma had a significantly increased expression of PSMA mRNA level as compared to benign nevi. And that was way more than what was than the upregulation in PSMA mRNA that you see in the transition from normal prostate cells to prostate cancer. So I thought that was pretty interesting. Then we did an immunofluorescent co-labeling study, and we showed again that PSMA is expressed in the blood vessels. The CD31 staining is specific for endothelial cells. And so when you see both colors on the same cell, you know it's an endothelial cell that's expressing the PSMA. And this image was actually given to us by the medical oncologist Michael Morris from Memorial, and it's an indium-11 J591 planar imaging, and it shows metastatic sites of melanoma when targeting neovessels. And I've seen, well, it's all over the literature too, that we can do this with PET-CT as well. So I had this project, I had this poster, and I had a late-breaking abstract presentation on PSMA expression in melanoma that was accepted. And I went to present my work, and one of the members of the audience actually said something to me that really, it impressed me. And he said, you know, this is a really exciting new potential treatment option for men with melanoma. And I was just like, wait, why men? And he said, well, don't you need a prostate to express this target on your melanoma? And I said, no. But I realized there was a problem here. It led to confusion. We were using different names throughout the literature, and this target, or this theragnostic target, really wasn't specific for the prostate. And so I published a paper in the Red Journal in 2018, and I proposed that we actually consolidate the nomenclature to folate1, which is also the name of the gene that encodes PSMA. A few months actually after I published this paper, I received news that my mother had breast cancer. And so this is a copy of her core biopsy of her right breast, and you could see that it's a HER2 positive invasive ductal carcinoma. Basically, I received a call from her radiologist that said she had a Ramirez-Fort breast lesion, spiculated right breast mass, and she also had this 1 centimeter lesion in her brain. So we did an FDG PET-CT, and it wasn't avid for her breast lesion or anything else suspicious. A month before my mother was diagnosed, this paper had come out, and it basically said that HER2 subtype tumors showed the highest incidence of brain metastases, and that patients with only brain metastases had a median survival of 17 months. I mean, this was for me a Darwinistic crisis. Then somehow those results led to tumor board saying basically, well, she has a small primary tumor, a low proliferative index, and it's really unlikely to metastasize. So we recommend that you do a craniotomy to evaluate what that lesion is that's radiographically consistent with a metastasis. And I just politely declined. So I was challenged with the kind of the same situation that happened during World War II, right? I had to think about cellular to organismal death versus survival. This data hadn't been published yet, but I knew that there was a survival benefit with aggressively treating these metastases if she only had one or just a couple more. And so I really wanted to know, is this a brain met, or are these tumor boards correct and this is something else? So I started wondering, is there any other diagnostic option that does not involve a craniotomy? And then I remembered, when I saw that video when I was doing the induction of PSMA in endothelial cell study, and when I learned it and I saw the immunofluorescence and we were targeting PSMA, I kind of saw or was introduced to PSMA PET CTs at the same time. So I kind of saw them as similar or parallel processes. And so it kind of clicked. And I said, all right, we need to get immunohistochemistry of my mom's tumor for PSMA. I requested this from the surgeon, and she was able to add it on to the staining. Voila. Then I started asking and calling centers. And believe it or not, even though this is 2018, it wasn't FDA approved yet, it was readily available, the radiopharmaceuticals, to be able to image PSMA with PET. And every center said no. And then I started reaching out to international centers and they also said no. So I thought, okay, well, all right, let's think of something else. Is there something else? And then I thought, wait, there's pertuzumab and trastuzumab. Can we use those antibodies similar to J591 and conduct PET CTs with those? At that point, I was put in contact with Gary Uhliner at Memorial. Well, he was at Memorial at that time. We were able to develop a protocol for a HER2 PET CT for my mother. And this is the famous picture that we shared at Capitol Hill Day. This picture shows, so on the left, we see the MRI of the 1 centimeter lesion in her brain. And then with her zirconium-89 pertuzumab, we conducted the PET CT and we could see that the lesion lights up very nicely and very little background. That led to my mom receiving aggressive local therapy to her breast cancer. She had a lumpectomy and lymph node dissection. And then she received gamma knife SRS to the brain metastasis. Then she started 4-agent chemotherapy with immunotherapy, so Taxotere, carboplatin, Herceptin, and Perjeta. That's trastuzumab and pertuzumab. Then she received whole breast radiation followed by a electron beam boost to her tumor bed. So my will to cure my mom ended up affecting all of my active research. And at every point along my subsequent research, I was constantly thinking of, okay, if my mom develops metastatic disease, like how are we going to cure her? How am I going to make sure that she doesn't die from this, right? I was challenged with death versus survival, and I picked survival. So I started another project beyond melanoma, and we looked at Merkel cell carcinoma. So we validated folate1 as a novel theranostic target in Merkel cell carcinoma. And specifically, we looked at 81 Merkel cell tumors and found that 60 to 77% of the patients expressed folate1 in the carcinoma-associated neovessels. And so this is just the immunohistochemistry. This was the isotype control. Here is CD31 staining, and here is the folate1 staining. And so we also found that Merkel cell carcinoma expressed significantly more folate1 as compared to healthy skin. And then we did immunofluorescent co-labeling, just like we did for melanoma. and confirmed that FOLH1 expression is restricted to the Merkel cell neovessels. But again, that threat to survival, that Darwinistic crisis where I was battling with my mother's diagnosis and had to choose, okay, death or survival. And again, I picked survival. My mom's tumors neovessels also express FOLH1. This is a slide I was saying. I took a second look and I said, this kind of looks like a brachytherapy catheter. And then I said, well, based on this concept, if the group of cells were that internalize the therapeutic isotope were to be considered as dwell points, just like we would for catheter-based brachytherapy, could we do radiation treatment planning for molecular brachytherapy using the same processes with the Monte Carlo simulation software that we would use for radiation therapy planning of catheter-based brachytherapy? So I showed in that publication that we could superimpose a Monte Carlo simulation onto the immunohistochemistry and actually map out electron emissions from a single point and see the ionizations in that tumor. And so where you see the yellow overlapping the blue, so the blue is a cell nuclei. Why that's important is because the yellow on the blue is where you would potentially receive or have a double-stranded break. And that would mean a mitotic or interphase cell death, which could potentially be a cure. So why is a Monte Carlo simulation? We use it because it's the most accurate method of modeling stochastic nature of interactions of radiation in matter, and it enables the characterization of complicated radiation environments for which dosimetry measurements are not yet available, like biopharmaceuticals. The simulation can apply a statistical sampling method to sample particle type, direction, the production of secondary particles using well-known statistical distributions and the physics properties or processes of electrons and photons of different energies. And so how would we do microdosimetry for radiopharmaceuticals? Well, we would need to establish a simulation geometry that would be made up of a mass geometry that's established from the CT scan and then the Hounsfield units, and then a virtual geometry, which is the contoured volume from the FOLH1 PET CT. And then we would need to determine source positions. And so here's that image that I kind of glossed over above. This is the prostate brachytherapy seed. And then this down here is the immunohistochemistry of a tumor expressing, those are little FOLH1 positive blood vessels. And to me, it kind of looked the same. And so if we consider that these are prostate brachytherapy seeds, we can kind of use that same distribution for calculating, I guess, dose distribution in a tumor that expresses PSMA in the neovessels only. Similarly, if it were a tumor that expressed PSMA on the cells, then we would expect the neovessel to express PSMA and take up the radioisotope and also diffuse along around this area of the normoxic cells. And then we can calculate based on the size what would be the expected cell number. And it's already known how many PSMA molecules are on link cap cells, which is kind of the gold standard for PSMA expression. So we can calculate dose that way as well. So how do we get an absorbed dose in gray? For there to be a dose, it requires the deposition of energy from a particle interaction with the mass geometry. So that's it with the CT scan within the virtual geometry, which is what we contoured. And then we can track initial decay, secondary particles, decay products of this decay chain throughout the simulated geometry until these entities come to rest or exit our geometry. And then dose can be accumulated on a scoring grid, which through the well-known fluence to dose conversion factors, they can be used to determine the dose in a region of interest that corresponds to the amount of radiation passing through the same region. These factors also consider the biological effectiveness. Again, that paper I published in 2018, since then, the FDA approved radiopharmaceuticals for PSMA targeting for molecular imaging and therapeutics, but we were still calling it PSMA. And I had that experience with my mother, and she was denied diagnostic opportunities. So I said, we have to do something about it, and I'm going to publish another paper on this, but in the context of how this misnomer creates gender and disease biases. All right, so again, the aliases are used interchangeably in the medical literature. Remember that paper I pointed out about planarian and PSMA? So that was one issue. But the continued use of PSMA, particularly in society guidelines, is really problematic because, first of all, for my mother to get the immunohistochemistry stain was an uphill battle. So that's a big problem. And it should be considered a misnomer because it falsely implies exclusive expression to the prostate. Also, this widely cited PSMA creates a gender bias that could translate into reduced diagnostic and therapeutic opportunities for genotypic female patients with folate1 expressing solid tumors. And the misleading nomenclature and related gender bias could adversely affect future FDA approvals for theragnostic indications for all solid tumors that express folate1 in the neovessels, including gynecological malignancies in women that do not have prostates. My mother was denied diagnostic opportunities. So it really is happening. And because of this, the United Nations Global Sustainable Development Goals and US General Services Administration content guide actually recommend using more gender inclusive language. And historically, the International Union of Biochemistry and Molecular Biology established that scientific nomenclature of enzymes should be based on the chemical reaction that they catalyze, because that's a unique feature of each enzyme. So folate hydrolase 1 uses water to break down glutamate residues from dietary folate. So this name, which is also the name of the gene, really describes the enzymatic function of, what everyone's calling PSMA. And we recommended that we establish a more gender and disease inclusive nomenclature by consolidating the name to FOLH1 as the gene that encodes the enzyme. So I do have a vision of the future of nuclear medicine, and I believe that really nuclear medicine physicians should be able to make a clinical decision choosing the radiopharmaceutical based on the expression profile of the tumor cells and not the gender or the disease of the patient. And why? It's because that theranostic scan that my mother had led to her being 6 years NED. And just like my mother, I believe that all women deserve the equal opportunity to survive.